Hey there, AP Bio, and welcome to our Chapter 13 discussion on DNA replication. So, before cells divide, we've been talking about the cell cycle with mitosis and meiosis, but before a cell can divide, we have to think, what is that first thing a cell needs to do to ensure that each daughter cell is going to be exactly the same? Think about what that is going to happen in that S phase of our cell cycle. We're going to need to divide our DNA. We need two sets of DNA. Otherwise, every time we divide, we're cutting our DNA in half, and eventually our cell's just not going to work anymore. So we need to remember that DNA is our hereditary molecule. It is structured to function as the blueprint for all of life. It has the instructions on how to make every single protein that makes up your body, that makes up who you are. So getting to this point, though, this double helix model and understanding how it divides, a lot of scientists go into it. A lot of times we think about those eureka moments and we read about one research project, but you need to remember that it's built on the shoulders of giants, and a lot of people contributed to that. We're going to talk about a few of them here. Frederick Griffith was a very famous experiment in the 1920s that suggested that there was some sort of molecular basis to inheritance. So we had already had Gregor Mendel's peas. We knew there was something causing hereditary. And so what Griffith did was he took these strains of a disease, both a non-virulent strain and then a dead virulent strain, and neither alone when they injected them. So if he put it into the non-virulent or non-destructive form of the disease into a mouse, nothing happened. When it was the killed form of the disease injected in, nothing happened. But when he mixed those two, the non-virulent and the virulent, together, it ended up killing the mouse. And so this suggested that there's something smaller than a bacteria that is conveying the instructions on how to become that virulent or disease-causing form of the disease. About 30 years later, we have Hershey and Chase, who were conclusively refining that work. And what they did was they used bacteriophages, which are viruses that infect bacteria, and they labeled these phages in order to infect cells. So what they were using is radioactive isotopes. And so first they used sulfur, which would be something that would be locked up in mainly proteins. And they injected that into the cells, and then when they centrifuged that, they found that the radioactivity or the phage protein was found in the liquid. When they did it with phosphorus, that got injected into the bacteria, and when they centrifuged it out, the radioactive portion wasn't in the liquid, but it was inside the cells, inside that pellet. And so what that meant is that the phosphorus was attached to the genetic material of the virus and not the sulfur. So that implied that it wasn't proteins, as originally thought were the genetic material, but it was DNA. Along came Chargaff, who showed the structure of DNA, that you're going to have an equal amount of adenine with thymine and an equal amount of guanine with cytosine. So those pairing rules, that A always goes with T, G always goes with C. And then using all this information, we arrive at Watson, Crick, Franklin, and Wilkins. So James Watson and Francis Crick published their study on DNA structure in 1953. And this was based on data that was actually generated by Rosalind Franklin, this famous Photo 51. And she was working in the same lab as this fellow here, Maurice Wilkins. Now, Rosalind Franklin was not working with James Watson and Francis Crick, and she actually published her manuscript at the same time, but it was not as well received as the Watson and Crick. People thought she was just confirming the results instead of inspiring them. So I'd love to have you watch this TED Ed video that I posted on Canvas. You'll also find the link in the description of this video that talks about the history of Rosalind Franklin, the discovery of DNA. It's a really interesting story filled with backstabbing and intrigue. So check it out. So we're here at nucleic acids, the basic for genetic inheritance. And we know there's two types of nucleic acids that we've talked about, DNA and RNA. And these are made of nucleotides. So remember that nucleotides have a pentose sugar, they have phosphate groups, and then they have a nitrogenous base. And so when we start looking at these, DNA is going to have thymine versus RNA's uracil, and DNA is going to be double-stranded, whereas RNA is only single-stranded. So DNA is the hereditary material that's going to get passed for genetics. And so we want to examine how this structure is built, because that's going to determine how our DNA is able to replicate. So those phosphate and sugars form the backbone, or the sides of the DNA molecule. Those sides of the ladder are going to be bonded by covalent bonds between the 3' carbon of a sugar to the phosphate of the next nucleotide. And we're going to be talking about what that 3' and 5' means here in just a second. The rungs of the ladder, those steps as you go through the DNA, those are connected by hydrogen bonds between the A and T and the G and C. So those nitrogenous bases are connected using hydrogen bonds. So remember, covalent bonds are a whole lot stronger than hydrogen bonds. So when we start replicating, those hydrogen bonds are going to break a lot easier than those covalent bonds will. So I mentioned 5' and 3'. So when we look at the structure of our sugar here, each one of these points is a carbon. So there's 1, 2, 3, 4, 5 carbons. And if we start from the O and go clockwise, you can count them. And what these 5' and 3' means is just the numbers of the carbons. So we can keep track of which carbon things are attaching to. So if we start at the oxygen and go clockwise, that's carbon 1', 2', 3', 4', 5'. So we can see that this is the 5' end where that carbon sticks off of the ring. And where the phosphate is attaching here is the 3' end of our sugar. It's the same thing when it's reversed here, because remember, our strand is anti-parallel. So here we're going from bottom up, 3' to 5', and here we're going from 5' to 3'. Same thing here. If we start at the O and go clockwise, that's 1', 2', 3'. We can see this is the 3' end, 4', and our carbon sticking off is our 5'. So always remember, if you're counting the carbon, start at O and just move clockwise. So there's 3 theories of how replication works. So do we just copy the whole thing and make a brand new strand? Do we break apart and use each strand as a template? So each new strand is going to have 1 strand of old DNA, 1 strand of new, or dispersive, where it's just a whole bunch of random cuts and copies. This one sounds pretty difficult. So it's probably going to be one of these two. And yeah, it's going to be semi-conservative. What we're going to do is break apart those hydrogen bonds to open up our strand, and we're going to use each strand of DNA as a template to build off of. We're going to just basically copy it, and that makes it really easy to build because A's can only bond with T's, G's can only bond with C's, so we just attach the nucleotides we need. So what I'd like for you to do is watch this video from Professor Dave, because it's going to have animations way better than what I can make. And I'm posting it to Canvas, and it'll also be linked in the description. And it's a great description of everything you need to know about how this process works. And I'll summarize it here. So it's important to note that this doesn't happen just in one place. There's going to be many different origins of replication, or where it opens up. And it's going to run in 2 different directions because the DNA is anti-parallel. And this is going to make DNA replication go a lot faster. Remember, you have millions of base pairs. So if you were just doing 1 at a time, it would take forever. So in our replication fork, as you saw in Professor Dave's video, there's a few enzymes we need to know. Topoisomerase, helicase, DNA primase, DNA ligase, and DNA polymerase 1 and 3. And then you're going to see there's things called Okazaki fragments, and we're going to have a leading strand and a lagging strand. So let's break some of those down. So to start off, helicase is going to break open the hydrogen bonds. It separates the hydrogen bonds, breaking open the two strands, allowing our DNA to unwind. Topoisomerase goes just ahead of helicase, and because as you're opening up, that can create a lot of stress and torsion forces on your DNA, it's going to break the DNA up ahead, unwind it, and reattach it so our DNA just doesn't explode and break apart. Once our DNA strands are separated, we're going to have primase, which puts down a little primer to act as a starting point for replication. Here you can see primase is putting down a little primer, and that's because we can't start copying without some starting blocks of where to start building. DNA polymerase 3 is going to start building the new strand, and it is going to build in the 5' to 3' direction. So on the 3 to 5 strand, because it's building anti-parallelly, that'll be our leading strand. It just follows helicase. But because our lagging strand runs the opposite direction, we have to keep laying down primer. So we'll put down a primer, build backwards, and then as helicase opens up more DNA, we'll put down a primer, build backwards, put down a primer, build backwards. And those little chunks that we build on our lagging strand are called Okazaki fragments. Finally, DNA polymerase 1 will come through and replace those RNA primers with DNA, and ligase is our glue stick. It's going to come together and seal all those little fragments into one fluid piece of DNA. So here we can kind of see that all put together. So we have DNA polymerase 3 and DNA polymerase 1, which are our builders. They can only build in that 5' to 3' direction. And that's why we have a leading and a lagging strand, but they do make mistakes during and that need some proofreading. Why don't we have a DNA polymerase 2? Well, we're not sure it exists. There's theoretically some in bacteria, but we haven't really conclusively found it. Remember, those Okazaki fragments are only on the lagging strand, and that's because as more DNA gets open, we have to build and go backwards, build and go backwards. And then all those mistakes that happened, we have something called nuclease, which cuts out damaged DNA, and then DNA polymerase will come in and replace it. And we also have things called telomeres, which are repetitive DNA at the end of the chromosomes that protect, because we're probably going to lose a little bit each time we copy. And so these telomeres mean we don't lose anything important. So here's a nice little example of how this all looks in action. I'll let it restart here. Our Okazaki fragments, our ligase is going to come in and seal them together. So, in summary, we're going to have DNA. Helicase is going to break those hydrogen bonds. Primase sets down a primer made of RNA. DNA polymerase 3 is going to start building in that 5' to 3' direction. On the leading strand, it just follows the leader, but on the lagging strand, because it's going backwards, we have to, as more DNA opens, put down a new primer, and then build backwards until we reach the old primer. Put down a new primer, work backwards until we reach the old primer. Polymerase 1 will replace those RNA primers with DNA, and then ligase is our glue stick that seals everything together to make it a brand new fluid strand of DNA. That's all I have for you for DNA replication. Make sure you know those major enzymes of DNA replication. Those are going to be really important. Understanding the 5' to 3' anti-parallel nature of DNA is also going to be very important for you to understand this process, and understanding that our covalent bonds in the backbone of the DNA stays strong, whereas the hydrogen bonds between those nitrogenous bases gets broken open. All right,